Okay, so today's topic is human embryology. It is a big, complicated topic and long one. So we will be dividing all this topic into two videos, and we will try to understand this all process with reasonable amount of information. In this video, we'll be discussing one phase, one most crucial phase of embryological development. So let's start. Embryological development is actually basically it is a development of an embryo into a fetus and a child. So it starts when a sperm fertilizes an egg and makes a zygote. It ends when a child is born. So usually it extends up to 9 months, though there are exceptions, we know. So we can divide embryological development into three phases. The first phase is pre-embryonic phase. Pre-embryonic phase, this lasts up to 3 weeks, starts from first day and lasts up to 3 weeks. In some books it's like 2 weeks, in other books it's 3 weeks. I basically consider it's 3 weeks. So pre-embryonic phase is first 3 weeks. Second phase is embryonic phase. Embryonic phase, it starts from the fourth week and goes up to eighth week. So there are 5 weeks: 4, 5, 6, 7, and 8. The next phase is fetal phase. In that, fetus develops, and it starts from the ninth week onward until the birth of a child. So we will discuss all these phases one by one. So let's start with pre-embryonic phase. Pre-embryonic phase starts with the fertilization. When, let me show you, when a sperm swims up into the fallopian tube and goes there, and the other side, the egg is secreted by an ovary, and here when a sperm meets with an egg, it's fertilized, and after the fertilization, zygote is formed as it's shown here. Then zygote will start dividing. There will be 4 cell stage, and then after 3 days a structure will be made which is called morula. What is special about this morula, this structure, that it has interconnecting structures between the cells. Okay, so morula will be formed on third day, then it will convert into another structure which is called blastocyst. On the fourth day, the blastocyst, the form, it is called early blastocyst because it will mature with the passage of time, and then by the sixth day, this will be a matured blastocyst, and we can call it, this is implanting blastocyst because it is ready to implant. So by the time when blastocyst is mature and day is 6, and this whole structure will keep moving along the fallopian tube and will come to the uterus and will attach implant to the uterine wall. So now if we summarize this first week, it consists of fertilization, formation of morula, formation of blastocyst, and implantation of blastocyst onto the endometrium of the uterus. Now let's see what is happening in second week. Second week, before we go to the second week, we must first have basic understanding of some structures which are important for an embryo to develop. That is why it is called pre-embryonic phase, because embryo is not still developing, but the structures which are essential for embryo to survive are being made. So in second week, some structure will be made. I just give you an understanding of those structures. What are those structures? These are sacs or cavities and villi. Here we have two embryos. This is reptile or bird's embryo, and this is mammal's embryo. If you just see here that this is embryo, and it is covered by a cavity which is called amniotic sac or amnion. As the reptiles and birds, they develop in the egg, inside an egg. They don't develop inside the womb of a mother, so that is why they need food in separate sac or cavity for themselves to nourish. So they have a special sac cavity which is called yolk sac, which provides them the nourishment, the food, whatever they need to grow. And if they're consuming something, so they must be excreting some wastage also. So there must be another sac for accumulating this all wastage. So here is a sac which is called allantois. All the waste which is being secreted by this embryo comes over here and accumulates here. So by now we talked about three sacs. First is amniotic sac, yolk sac, and allantois or allantoic sac. And there is a fourth sac which is the bigger one, and this circular line you can see, this is sac. This is called chorion or chorionic sac. This cover all these three sacs. So we have discussed four sacs: yolk sac, allantois sac, amniotic sac, and chorionic sac. This was the embryo of reptile and a bird. Now let's move to the mammal. In mammals, the bigger sac, what we can see here around the embryo, this sac is amniotic sac, because there are the other sacs, but they are smaller in size. Though these other sacs are present, but they are collapsed. As we can see here, this sac is yolk sac, but it is much smaller than this one. Why? What is the reason? Because in mammals, the embryo will be getting all the nourishment from the mother, from mother's bloodstream. And allantois sac, which is this one, this is also small in comparison with this sac of the reptile or bird. Why? Because the waste product which is produced by the embryo will be given to the mother, and mother will take care of it. So these two sacs are kinda non-functional, though these sacs have some functions, but we will not be touching those functions right now. For our purpose, these sacs in mammal are non-functional. So there are yolk sac, allantois sac, this is amniotic sac or amnion, and there is a chorionic sac, chorion. So four sacs. So if this embryo in the mammals is not getting food from yolk sac and is not secreting its wastage into the allantoic sac, so it must be having some structures which will help it to get some food from the mother. So these are the structures. These are the villi, finger-like projections, which are made from chorionic sac here. So these projections are full of blood, the blood of this fetus, of this embryo. So on the other side of this villi will be the blood of mother. So one thing what I want to mention, the mother's blood will not merge with fetus blood. No, there will be close relationship. They will be close to each other. They will project close to each other, but they will not merge. They will not diffuse. The blood as you can see, these villi are blinded, so they are not fusing into the mother's blood vessels. So there is a connection between them, but they are not mixing, fusing with each other. So this whole part and all these villi will make umbilical cord, which further on in later stages we will see will help embryo to get all the nourishment. Now we have the basic understanding of the sacs and the embryonic, chorionic villi. Let me show you one more pic, because these structures are very important to understand the next week what is happening. Here in this pic, we can see we took a cross section from this place. This is a fetus. So if we see here, so there are these villi, chorionic villi, and these are the blood vessels of the mother. This whole part is called placenta, placental section. This is placenta. We took a section of placenta. These are the mother's blood vessels. These are the villi. This is umbilical cord coming. So here is a baby attached, as we can see in this structure here in this pic. Now let's move on. Let me show you one more diagram here, which will firm our understanding. Yes. This is the endometrium. Okay. This is embryo. These are chorionic villi. This is a yolk sac. This is amniotic cavity or sac containing amniotic fluid. And these villi are made from chorionic sac. So there is a lentoid sac as well. So we can find all four sacs: a lentoid sac, amniotic cavity or sac, yolk sac, and the outer side, this is chorionic sac. So here, these villi, along with the blood vessels of mother, are making umbilical cord. So this is umbilical cord coming out. What is this? This is an embryo. Now let me show you another pic, but which will show you that how the embryo will develop in second week. In second week, the basic structure which will be made are chorionic villi, because a placental relationship will be made in second week. Here in this diagram I drew myself, there's a blue, red, and green. These sections, these layers, these indicate an embryo. So in second week, embryo makes 3 layers, just similar to 3 papers placed on each other. The upper or outer portion is called ectoderm. The inner portion or inner layer is called endoderm, and between these 2 layers there is a layer which is called mesoderm. So there are 3 layers here. This is amniotic sac, this is yolk sac or gut, and this is, you can say, this is a space between chorionic sac and these structures. These are coming villi. So there are few terminologies what I must mention here. This portion is called decidua basalis. This portion of placenta is called decidua basalis, where the chorionic villi meet with the blood vessels of mother. The opposite side is called decidua capsularis. And the point where these villi are coming out, this is called chorion frondosum. And opposite side, it is called chorion levi. So decidua basalis is a part of a placenta where the villi are projecting to the blood vessels of mother. So this is decidua basalis. The opposite side of decidua basalis is decidua capsularis, and where the villi are coming out, this is called chorion frondosum. The opposite side is chorion levi. So these terms are important because these can be used in MCQs. So as a side note, this is a time, the second week is a time when amniocentesis is done. So his question comes, what is amniocentesis? Amniocentesis is a procedure or a method of getting amniotic fluid from amniotic sac of developing embryo. So a needle is inserted through the abdomen of the female, which further goes to the uterus, crosses the amniotic sac, and takes a sample of amniotic fluid and comes out. So this amniotic fluid is used to analyze and check either this embryo later on in its life will be suffering some genetic disorder or not, like Down syndrome. So amniocentesis is a bit risky. So nowadays another procedure is done, which is called chorionic villus sampling. Chorionic villus sampling, CVS, in this procedure, tools are inserted through the vagina of a female and some microscopic section of chorionic sac or boundary is taken. It can be some section from the villi, or it can be anywhere from the chorionic boundary. So let's summarize what happened in second week. In second week, embryo is developed into 3 layers. Outer layer, ectoderm. Inner layer is endoderm, and between them there is a layer which is called mesoderm, and placental relationship is made through these chorionic villi. So these two very important structures are made. Now if we move to the third week, what is happening in third week? So before I tell you, let's see. Let's just consider that these are 3 layers. This is ectoderm, this is mesoderm, this is endoderm. These layers are on each other. What actually happened in third week? These layers fold together. This was ectoderm, this was mesoderm, this was endoderm. They fold together ventrally from front. Okay, so there is a peak. Got a structure here. So they fold together, but here it will be made head. They don't fold completely from the upper side, and they don't fold completely from the down side. The last point where it folds, these layers fold, is your navel, your belly button. And this is day 22, and this is day 28. Then spina bifida, this is a condition when these folds don't close very well. So we are not discussing this condition, but what I want to show you, this is a way when these folds will fold. These layers will fold. So in third week, these layers will fold and make further some structures. Ectoderm, the outer layer, will make neural tube. Later in this development, we will see, not in third week, later in the development, this neural tube will make central nervous system. Ectoderm will also make surface ectoderm. Later on, this surface ectoderm will make epidermis, the outer part of the skin. So ectoderm is making the skin. So this is the outer part of our body. Mesoderm, the in-between layer, it will form three structures: paraxial mesoderm, so intermediate mesoderm, and lateral plate. The paraxial mesoderm, para means around or about, axial means the center. So this mesoderm, this section of mesoderm is in the center. And lateral plate, lateral means periphery or the sides. So lateral plate mesoderm is on the sides. In these two, between these two mesoderms, there is intermediate mesoderm, which is in between these two. So what structures are made? From ectoderm, neural tube is made and surface ectoderm is made. Central nervous system and epidermis will be made, but later on in the embryological development, not in third week. Mesoderm will be further divided into three different sections: paraxial in the center, lateral, that is on the periphery, and intermediate in between. Later in the development, not now, the lateral plate mesoderm will further subdivide into somatic mesoderm and splanchic mesoderm. We will see what they will do, what is their function, but I just wanted to mention in third week, the endoderm will start forming alimentary canal, your gut, digestive tract. And one more thing, as a side note, which I want to mention, that chorionic gonadotropin hormone is a hormone which is secreted by chorionic sac around the embryo. And this starts secreting in third week. And this hormone is important in sense that it will stimulate ovaries to keep producing progesterone, because progesterone is important for the vascularization, for the blood vessels in the placenta to be made. And with the passage of time, the blood vessels will be more and more, placenta will get thicker and thicker. So this all is because of this hormone, progesterone, and progesterone is secreted by ovary. And what hormone is stimulating the ovary to keep doing that? That is chorion gonadotropin, or CG. Okay, so this what is happening in third week. And this hormone is very important in that sense also, that it can be used to identify either the woman is pregnant or not. So there's a test which will confirm the presence of CG. If there is CG, so there will be a change of the color in the stripe, which is usually used for pregnancy test. So let's summarize once again all these three phases, because we will be covering only pre-embryonic phase in this video. What happens in the first week fertilization, number 1. Number 2 is ampulla formation. Number 3 is blastocyst formation. Number 4, implantation. What happens in second week? Second week is placental relationship is made through the chorionic villi, and placenta starts developing, and 3 layers of embryo of this embryo are made: ectoderm, mesoderm, and endoderm. Number 3, the third week, the neural tube is made from the ectoderm, and surface ectoderm is start being made. And mesoderm will be further divided into sections: paraxial mesoderm, intermediate mesoderm, lateral mesoderm, and we will see how these mesoderms will develop into further structures, because eventually they will be making some organs in the child's body. And endoderm is making alimentary canal, which is gut. So that's up for today. Thank you for watching. Bye bye.